I have the pleasure of bringing to you today a video first in a series on Asian sea bass, otherwise known as Barramundi. So in the first video, I'm talking here about production and biology, and then a second part, I'm going to be talking about nutrition. Well, of course, in the future, I will be adding more parts, but at the moment, we have these two. So please enjoy the first one. Here we go. So, part one, production and biology. And this is a talk that is based on a presentation that I gave at Aqua India in Kochi, India in the early beginning of 2020. And in fact, a few weeks after I went to India to give this talk, then the entire region shut down due to the arrival of the COVID-19 pandemic. So as I mentioned in the introduction, we have the Asian sea bass, otherwise known as the Barramundi, which is an Aboriginal Australian name, but it's very commonly referred to as the giant perch. So what I have here for you is a page screenshot from Seafood Watch. So this is a website set up by the Monterey Bay Aquarium in California, and it gives recommendations to fish that consumers should feel confident in buying and which fish they should avoid. So we see here that it says giant perch, and the species is Lates calcarifer, and it's also known as cockup, giant sea perch, coral, narefish, palmer, and silver barramundi. And it's available more or less worldwide. Now, one of the things that Seafood Watch does is they rank the source according to its environmental impact. So we see here that the best choice is indoor recirculating tanks with wastewater treatment. And then good alternative, is farming in marine net pens. And then the third option is indoor recirculating tanks without wastewater treatment. So when we then talk about growing in marine net pens, they also begin to distinguish whether or not we're talking about fish that are grown with manufactured feeds, which is a better alternative than feeding with uh, forage fish, trash fish, or whatever you want to call it. In general, they suggest that we should avoid fish that are being grown and using fish as feed. So I've talked about fish.j before, and I find it's a very useful place to get the latest information about production volumes and values. And so I looked up Asian sea bass in the workspace that you can see at the bottom, Global Fishery and Production Statistics to version 2024, which gives us numbers as recently as 2022. So we see in 2015, production volume was around 80,000 tons, and it has increased to around 154,000 tons. And we look at production value, $328 million in 2015, and now approximately 600 million US dollars in 2022. Now, there is a lot of concern about overfishing, and there's a lot of interest in finding alternatives to wild sea fish for consumption. And so there was a lot of interest expressed in around 2005 and about 2006 when the Australian company Australis Aquaculture started a marketing campaign to promote wrasse-reared Asian sea bass. So we have this article from the Boston Globe magazine from 2006 calling Asian sea bass barramundi the next big fish, coming soon to a plate near you. And then they had a chef report that it it's an excellent fish, very versatile in the way it handles various cooking techniques. Because it's farm raised, it's available year round, and not many wild fish are nowadays. And then at the same time, actually a little bit earlier, there was this article that showed up in Intrafish, um, and the title of that article was Barramundi, a fish whose time has come. And then they mentioned two companies, one of which was Australis, that they had raised nearly 12 million US dollars to expand their barramundi farming operations. Now, if we're talking about cage farming as is done in Vietnam, it's not necessarily an expensive proposition, but if we're talking about growing these fish in recirculating aquaculture, it tends to be quite expensive. And so it requires quite a considerable investment, and it's actually quite risky. Nevertheless, the key point of this slide is to just show you that there is a lot of attention being paid to Asian sea bass, and I'm assuming that it will have a very bright future, especially as it is very adaptable to farming where many other marine species are not. Okay, so for the biology, this fish, while we typically will see it in a market maybe at a 2 kilogram to 4 kilogram size, in the wild, it has been observed to be as long as 2 meters, 200 centimeters. And it reaches an average length at sexual maturity of about 90 centimeters. Now I have this picture here on the left-hand side showing you above we have a male fish and below we have a female fish. So the female fish tends to be for the same size, it tends to be a little bit heavier. And if you notice that the depth of the body tends to be deeper. And there are some changes if you look at the mouth area. The male has a much more aggressive mouth than the female seems to have. On the right-hand side are some pictures of farmed Asian sea bass from Cox's Bazar in Bangladesh. And you can see in that upper picture that the fish actually has quite a small head relative to its body, and it also has quite a large mouth, which is not so apparent if you're looking at the pictures on the left-hand side. This fish then is a very aggressive carnivore. Talking a moment about its ecology and environment. One of the things that makes the Asian sea bass very adaptable to aquaculture, it is a euryhaline catadromous species. So euryhaline means that it is very adaptable to seawater of really any kind. Adults live in freshwater, but are capable of living in brackish water and full strength seawater. Now, there are Asian sea bass farms at the north coast of Bali in Indonesia where the Asian sea bass are being grown in full strength seawater. And the texture of the meat is very firm. It tastes just like a marine fish. Where here in Thailand, we tend to have pond raised, farmed Asian sea bass, and the texture and the taste are a little bit different. So we have this graphic over here on the side, and it just kind of shows you the migration pattern of the Asian sea bass. We have the fish spawning in the sea, salinity 30 to 32 ppt. The eggs drift, and then the larvae develop, and then the larvae hatch, and you have the coastline, the inshore areas are the nursing grounds, and the salinity is typically a little bit lower, especially if you have some fresh water coming into the sea nearby. Then, as the fish grow, they migrate upstream, and then the feeding ground for post-juveniles is in brackish water or freshwater bodies. And we have seen Asian sea bass in Thailand um, living essentially wild, uh, 100 kilometers or maybe even 150 kilometers inland from the sea. So here we have a map showing the territorial range and frequency of Asian sea bass all across the Asian Pacific region. And according to Fish.j, on the right-hand side, you see the list from their database of all of the countries that have reported growing Asian sea bass in, let's say, the last 10 years or so. And I left out some countries that maybe had experimented with them in the past, but they do not do it at present. What we see with the Asian sea bass, and when we talk about frequency, where you see yellow, this means the fish is present, but not necessarily in high density. And the areas that are bright red are places where the Asian sea bass is very commonly found, and it's regularly there. And so we see it stretches all the way from the warm southern waters of Japan along through Jeju, South Korea. It goes down through around the coast of China, around Taiwan, all the way down through Southeast Asia, down to the northern Australian, essentially tropical, semi-tropical coastline, and then across India through the Seychelles Islands, and then over close to the African coast. And they're even showing some here in the Saudi Peninsula. One of the things that, in mentioning that the fish is considered to be a good choice because it can be farmed, we see here this data is coming from another website called Fish Choice, where they also provide information. But this is typically more a business to business website where they provide information to seafood retailers rather than consumers. And what they're suggesting here is when they compare wild fish, the main supplier of wild Asian sea bass has been Indonesia. And this is not really a recommended source because wild landings have decreased 15% in 2016 compared to only 2 years earlier in 2014. So it is a fish that the market wants to see coming from farms. So looking at where the fish come from, we see 35% from Malaysia. Now, back in the day, they used to feed fresh fish. Now they're mostly using feeds. Um, in Indonesia today, a lot of farmed Asian sea bass is happening, and they're also farming a lot of grouper there, but they have mostly converted now to feeds for the farmed fish. We see Thailand here supplying about 25% of the harvest, and then we have Taiwan, Indonesia, Saudi Arabia, Australia, and right there at the very bottom, the best choice that they support is the Vietnamese grown cage, ocean cage farmed Asian sea bass from Australis company at that time. So another biological characteristic of Asian sea bass is that they are hermaphroditic. And hermaphroditic means that the fish has the ability to essentially change its sex and change its phenotype, meaning that it can go from being a fertile male to becoming a fully fertile female. Fish generally start out more as males, and they may spawn once or twice as males. Then at larger sizes, they revert to females. And I've given you here a paper called Early Sex Change of Barramundi lateus calcarifer, farmed in Singapore. Another paper here, I have not given you the link at this point, but I believe I have given it to you. We'll give it to you later. We see that the gonadal maturation stages in Barramundi are as classified by this author, that we have male, and then we see the development through the different stages, M1, M2, M3, M4. And then at this transitional stage, the testes begin to degenerate, and we see the beginning of the female ovaries instead. And then finally, at the end, we see full development of egg producing uh, uh, ovaries. This paper I mentioned on the previous slide, Early Sex Change of Barramundi, what they noticed was that when their fish were becoming female, that they were becoming female at earlier sizes, earlier ages, than fish coming from other regions. And so the color code for this particular graph here, and for this graph you can see here. So we have SG is Singapore, FP is French Polynesia. So that's like Tahiti. And we have male Singapore, male Polynesia. We have the trans colors, and then the dark purple and so on here is the French Polynesia, but female. Similar code, but not quite as clearly laid out. We see in this data here from Singapore. Note that when they compare the blue, which is Singapore, with the other fish don't turn to be female until about here, but at Singapore, they're turning to be female, starting at about here. It becomes quite obvious that most of the fish are in transition, and this is a transition here, and these are fully female. And that's occurring at about 4.5 kilos weight, whereas with other areas like in French Polynesia, it's occurring at around 6.5 to 7 kilograms weight. Then they talk about the fact, well, how does this work? Because the genes are the same. The genes inside the body, inside the cells, do not change. And this sex change that occurs in these fish is apparently related to epigenetic modifications. And they list here several epigenetic mechanisms that have been determined. And here it tells us where in the testis and in the testis, and then in both males and females. And this is another paper if you want to learn lot more about the sex change that occurs in actually many species of fish, you can scan this and find an interesting paper to read right there. This is some data from Australia. I will give you the reference to this later. And we can see here that the average daily gain is 5.6 grams per day. Now, this is coming from a Asian sea bass farming manual. We don't necessarily know much details about the feed. We don't necessarily know much other than it says that these are in marine ponds. We don't know the state of genetic development or domestication of these. But the Asian sea bass farming, the Barramundi farming in Australia is not a huge industry. But nevertheless, I would say that we could expect 5 to 6.6 grams a day, but we might actually see some other regions that have more advanced techniques, maybe like Vietnam, or maybe growing the fish faster, but I'm not able to find any data about that. Now, this is a little bit old. This data is from Thailand, from uh, the paper was written in 2018, but the data actually came from considerably earlier than that. And it's coming from 4 different provinces, and we can see here that we're marked as 1 in the south, and then we have 2. So this is Champon, and then we have 2 over here. This is Chechen Sao. We have 3, which is Chonburi, and we have 4, which is Sumsongkhram. And this is actually where I live in Thailand, in this area. And there are a lot of Asian sea bass farms scattered here and there. Actually, they're mostly all former shrimp ponds. So what we see that there are some different strains. They identified 3 or 4 different strains in Thailand, and these are picked because these are somewhat different. And when we grow them, you see here the age up to say 210 days. Um, the growth is about like that. Now, I have not taken the trouble to calculate the average daily gain, but we do have the size and we have the days. We can make that calculation. Now I'd like to shift focus a little bit and talk about gut physiology. So moving here now away from production into biology. This is a picture from a paper published by Hermaphroditic et al. In 2020. And they're actually looking at bone structure in relation to mineral intake and vitamins and so on. And it's a wonderful X-ray, which I thought I would share with you here. And the paper actually has a number of other interesting photographs. So the Asian sea bass are opportunistic predators. They are very predatory. And if we look at this graphic, it would go from 4 millimeters up to 1,000 millimeters. And so that's 1 meter. And we see that at the very beginning that the diet is mostly microcrustacea, a small amount of macrocrustacea, but I think the problem here is that the size of these prey items are too large and not much is eaten. And then we have here other invertebrates. So other invertebrates, we assume then is not crustacea, so that can be maybe things like shellfish and other things. And then we see that as the fish grow and they are larger, they can eat larger sizes, that they begin to shift away uh, from microcrustacea into macrocrustacea. So this could be shrimp, it could be small crabs. And then we see a slow increase until at the end, most of their predatory food items are fish. I found this paper by uh, Purush Othman from 2016, published in Peer J, an open source journal. And he wrote several papers about the digestive gastrointestinal tract of the Asian sea bass. So I took this photograph from him, and we see on the right-hand side the extracted gut from the esophagus at A right here. So the esophagus is right here. We then have C and B are the stomach. We have G is the mid intestine. So this is the fore intestine, the mid intestine, the hind intestine. And then we have the rectum over here. So you can tell by the letters of the alphabet which part of the gut is being referred to here. And there's a ruler there so you can see the size. Now, I don't know what size this fish was that this gut was taken from, but I don't believe it was too big. Now, one of the things that we know is that as the diet changes, and we saw that from as the fish were larger, it moved away from crustaceans, micro feeds to macro feeds, and so on. We then have a development of the digestive tract, which kind of follows or prepares or even tracks the changes in diet. So this paper, Development of Digestive Tract and Proteolytic Enzyme Activity in Sea Bass, Larvae, and Juveniles, and we're looking here at pH. So in this graphic, we have a dotted line here, which is representing fish that are being fasted. So they are not being fed. And presumably at the beginning here in this stage, in the first 10 days or so, these could be yolk sac fry, and they have absorbed their yolk, and they are going to begin feeding somewhere in this area right here. So when we talk about feeding larval fish, here we're basically talking about feeding them things like rotifers, artemia, uh, and other small organisms that we grow and feed in hatcheries. So this line, the black line here, is representing fish that are being fed. And this line is representing fish that are being fasted. Now, fasted means that there's no food coming into the intestine. And so we basically see that the pH is more or less stable. And it is at around 7 to 8. So it's basically essentially a neutral pH because it's not doing anything. But in animals that are fed, where they have to digest the food, we see that the pH drops. And at around 30 days, the pH is leveling off at about 4. And so this is the level at which their stomach enzymes would be optimally functioning. Now, we also assume that over the time here, the food is changing, but the animal, typically as we eat like pelleted foods, the pH tends to rise. And so then the animal has to secrete more acid in order to keep the pH down. So we don't know exactly what these animals are eating, but we see that pH is actually quite a reasonable pH for effective protein digestion in the stomach and upper intestine. So here we see the foregut, midgut. So this is gut pH days after hatching. And we see once again, fed is the solid line and fasting is the dotted line. There's not a lot of difference. There's a slight increase in slope for the fasted. and a much lower increase in slope, more really we might also consider to be essentially unchanged at somewhere around pH 8. So what happens in the foregut is at the point where the stomach exits and the intestine begins. This is an area where we have pancreatic secretions, and the pancreatic secretions adjust the pH upwards and make it more alkaline. So that's why we see here in the foregut and midgut, and then also in the rectal area here, that the pH is more or less about the same and not particularly different between fed and unfed. This is a paper by some Indian researchers. This is actually the quality of the paper that I have. So it didn't look like a photocopied, but may have been. And so we see here some proteases that they found. They found chymotrypsin in the intestine and pyloric ceca. They found some elastase. They found leucine aminopeptidase and trypsin and some pepsin. Now, pepsin is an protease that's found in the esophagus and in the stomach mainly. And then when we see it in the intestine and so on, it's basically more or less just drifted out of the stomach and it will be diluted down over time. And as it moves through the gut, it will be less and less. It's either broken down or it's just diminishes. So there in the intestine, on the other hand, we see that we are going to be depending mostly on these aminopeptidases. These are the ones that break down peptides into free amino acids and short chain peptides, dipeptides, tripeptides, and they're mainly active in the intestine and the pyloric ceca here. Now, when we're talking about marine fish, we're often wondering about their ability to produce carbohydrates because dietarily they typically do not eat any carbohydrates, other than maybe a small amount of glycogen, which is coming in the muscle that they consume from their prey. Nevertheless, there seems to be the presence of some amylase, which is effective against starch and also against glycogen. Um, there was some maltase activity and trihylase activity, and then interestingly, they also found some chitinase activity in the stomach, and it's not particularly high. And this may be representative of the fact that the Asian sea bass is eating crustaceans. And the ability to produce some chitinase means that it can utilize some of the chitin that it's consuming with either insects or maybe with small crustaceans. Now, what we don't know from this work is the extent to which there may be gut microbiota contributing to the production of some of these carbohydrates enzymes. Here's a paper uh, done by this Thai researcher looking at the pH optimum of proteases, and he's looking at both of these serine peptidases. We have the chymotrypsin in the purple, and we have the trypsin in the red, and the pH optimum we see is somewhere around neutral, which is 8, as you can see here, is somewhere around 8. There is a bit of an earlier peak at around 5, as you can see here. So these are basically working primarily in the foregut, the upper intestine. So here the researchers now are looking at temperature optimums of the enzyme. So we see the red once again, chymotrypsin, and the yellow is trypsin. And surprisingly, it's showing that the maximum activity is going on at about 60 degrees Celsius. And it's often a puzzle why these enzymes seem to have their greatest activity at temperatures that are far above the biological limits of the animals. But if we go down and look at the range where the animals are comfortable, which is down here, maybe in not greater than about 40 degrees, we see that at this temperature, which might be a little bit low, digestibility might be a little bit slow. We seem to have a reaching some kind of a optimal level here that's quite consistent across several degrees increase in temperature. And so this is the area where the enzymes would work. Now, the thing is that an animal has a feedback system where, depending on the amount of protein in the diet, it will produce more enzymes. So there's nothing wrong with having this level of activity. The animal will simply produce more enzyme, and per unit of protein to be digested, it would have to make maybe double the amount of enzyme to do the job as it would maybe need at a much higher temperature. But of course, this is not a realistic temperature. So here's some work from Bermudez and Glenn Cross and another author, I don't remember. And they're looking here then at temperature along the bottom axis, and they're looking at growth rate in grams per day on the vertical axis. And they're looking at two sizes of fish, small at 20 grams and a little bit larger at 140 grams. We see that as the animal's temperature rises, this green area is their comfort zone. The maximum growth rate is really occurring at about the middle of this comfort zone. And then as the temperature continues to rise and exceeds their comfort zone, then we see a rapid decrease in growth rate. And this is basically being caused by a metabolic dysregulation. Um, the temperature of the enzymes and the hormones and everything else um, is basically kind of excessive. Everything is overactive, but not necessarily in sync very well. And so then we have problems with fish eating but not growing, and so on and so forth. I wanted to note here that these lines also have error bars. So this would be standard deviation around their treatments. We see at the beginning that the error bars are very small. You almost can't even see them. Here we're beginning to see some variation in the larger, but still very minimal variation in the small. And then inside the comfort zone, we see here that there is a fair difference. So this might be, plus or minus grams on 20 animals, 30 animals, or however many they may have used. Some grow faster, some grow slower. It's just the way biological organisms work. But as we exceed the comfortable temperature, notice that the error bars shrink again. So this is implying that every animal is being affected negatively and every animal is growing poorly. Some animals are not doing better than others. On the right-hand side, we see this three-dimensional graph where we have live weight across here going up to 250 grams, and we have temperature on this axis. And in the middle of this range, we seem to have the best growth rate at this point here for the larger animals. When we go below this temperature, growth rate drops. When we go above this temperature, growth rate drops. And this axis is the growth rate in weight gain per day. Okay, we know that this animal is a very predatory species. And in most farming situations, we do have considerable problems with cannibalism. A large Asian sea bass can eat a smaller fish 70% its size. So Ship has shown us here this photograph showing this Asian sea bass on the right eating this Asian sea bass on the left. And this may be the fish after it has been pulled out of the larger fish, showing that it's basically has suffered maybe some enzyme secretion damage. Basically, even if it were to escape, and it probably couldn't because these fish have very sharp teeth. Um, so large fish, 1.4 times larger than the small fish. So many factors can influence cannibalism. So we have stocking density is one, feeding regimen is another, and shelter and lighting is a third. So let's talk about those briefly. This paper is talking about that Asian sea bass, they grow them in low light conditions and they even grow them in darkness. So they have here a population grown in continuous light, but it's not necessarily a high light condition. It may be moderate to low lighting, but nevertheless, it's continuous. We look at the mean weight and we see it's between 20 and 25 grams. And there are a few large animals, 45 to 50 and 50 to 55 grams, maybe one or maybe two or three, not more than three. Whereas if we go and look at continuous darkness, the average weight is much more, um, let's say, standardized relative to the standard curve behind. And we basically don't see any organisms above 40 grams. Somehow, this suggests that cannibalism uh, is partly dependent on visibility and the ability to see. So in this situation, what's happened is these larger fish are eating the smaller ones, which is skewing this population. And they're picking animals about 70% their size, so they're taking fish out of this size. They're not interested in the small ones over here. But in this one, where they're growing in the dark, they are basically swimming around, but they're not really seeing one another. And so will still be some cannibalism, but very little in this situation. Now, this paper also talks about preventing cannibalism, but in this case, they're not only using low light, but they are also using refuges. And the refuge is basically a plexiglass box, which is just high enough to allow the small fish to get in. They have a stiff spine on on the back of their spine, and it's not very flexible. So this refuge was built just big enough so that they could fit in there. But the larger animals, and you see here it talks about the large animals were length 8.5 centimeter and the small animals were only 6. That's not a very big difference, but in terms of the size of the animal, it was enough. So the small animals could hide in the shelters, but the big animals could not get inside the shelter because they were designed not to fit the big fish. And then we have 4 lines here. We see NH is no refuge, but highlight, no refuge, low light, refuge with highlight, and refuge with low light. So this is our NL, NH, RL, and RH. And we're looking here at cumulative mortality. So bigger is worse. Uh, when we have both refuge and low light, we see typically somewhat lower than 30% mortality cumulatively. But for the ones under high light and low light, but no refuge, we see cannibalism went up to mortality went up to of the small fish went up to 100% very, very quickly. Over on the right-hand side, what we're looking at here is chasing frequency and biting frequency of larger fish on small ones. And so then you rank these as you can see here, NH and L. So those have no shelters, but high and low light. And these two have shelters. And the simple presence of the shelters means that the small fish are protected and they're not being chased and they're not being bit. Okay, so the thing is that even if one animal doesn't consume another, but it chases it and bites it, then you end up with skin wounds, you end up with infections, you end up with parasites, fungus, and various other things. And so you might not have these animals being killed outright, but they will be beaten down and become weak and easily susceptible to infection and disease. So just now we're talking about density, and I found this data from the FAO AFRIS database showing um, some information about fish size and stocking density. And noting here that this grower fish size 50 to 100 grams, 100 to 600, 600 to 1 kilo, and above 1 kilo, they're growing 15 to 40 kilograms per cubic meter. Feeding was floating or slow sinking. Whereas when we look at cage culture in Asia, and presumably this is not intensive, this says up here this is intensive. We see 40 individuals in Thailand, 44 in Singapore, 100 in Australia, 100 kilograms per cubic meter in Australia. Then we see individuals 15 to 25 per cubic meter in Australia, in Malaysia, 12 to 300 in Australia, and so on. And we see the stocking size. So then this will have a direct impact on the cannibalism because the higher the stocking density, the less easy it is to feed fish effectively. That is, every fish obtains the food that it needs. And so then you'll have a problem with cannibalism. How do we prevent cannibalism? Well, manage stocking density to minimize the stress response, uh, grade larger fish out and separate if necessary to maintain small size differentials. And remember, it said that the fish 1.4 times larger could eat a fish at the scale of 1. So that would require quite a bit of grading. Now that could be done with grading bars, it can be done with nets and so on. But feeding practices play a big part in it. So we need to do proper practices for weaning fry onto manufactured feeds so that all fish are converted simultaneously. Now, what I've been told is that there is a window of opportunity that if you try or too early, you might not succeed. But if you wait too long, the fish actually resist learning how to eat manufactured feed. So this is typically a job that is performed in an Asian sea bass hatchery where they train the fish to eat pelleted feeds before they sell them to the farmers. Another point, when the energy density of the feed is too low for fast growers, they look for food elsewhere. Likewise, if you underfeed, you have the larger fish, they typically would consume a greater volume of feed. They're hungry and they will look for something else and they will then start to cannibalize smaller fish. Researchers have looked at how many times to feed per day. It should be 3 to 4 times per day. uh, better than 1 time per day, and some reports suggested that 3 times was better than 4, but I'm not certain why that might be. And then we know with other species, we know with swine, for example, we know with grouper and a couple of other marine species that if we add some of the amino acid tryptophan to the feed, this has an impact on behavior through making some metabolic changes in the brain. It reduces aggressive activity and it reduces physiological stress response. Having these fish being aggressive carnivores has one benefit, and that is that Asian sea bass is often in Asia is often used in polyculture as a police fish. And I found this paper, Growth and Predator-Prey Interaction of Asian Sea Bass and a Species of Tilapia in a Polyculture System. So the researcher used a 1.1 hectare pond, and then they had 3,000 tilapia alone. They had 3,000 tilapia plus 600 Asian sea bass, so that's a ratio of 5 tilapia to 1 Asian sea bass. And then they had 5,000 tilapia to 600 Asian sea bass, so that was a ratio of 8.3 tilapia to 1 Asian sea bass. And then when we look at the data for the growth, we see the average body weight, we see survival, specific uh, uh, daily weight gain, we see the harvest biomass, the FCR, and that the presence of the Asian sea bass actually improved the profitability. Okay, so the fish farmer can sell both the tilapia but also sell the Asian sea bass. And when they did the cost benefit analysis, the T1 treatment with 5 tilapia to 1 Asian sea bass brought a 27% higher profit than the control. And the T2 treatment with 8.3 tilapia to Asian sea bass, 1 Asian sea bass brought an 86% higher profit. And then in the lower right there, I mentioned that there are some other researchers who have tested fish at different levels, 15 to 1, 20 to 1, and also lower at 5 to 1, as was in this experiment I just mentioned. So I'd like to close by giving you some resources that you can use to learn more about Asian sea bass farming and production. And probably the best one is the one on the left side here, Biology and Culture of Asian Sea Bass. Um, this was written by or edited by Dean Jerry, and surprisingly, he offers it as a free download even from his own personal website, and you can find this um, readily available to download from many places. Many of the papers that are in this book are referenced in Google Scholar, and when you click on them to download them, you actually end up downloading the entire book. Now on the other side here, we have training manual. This is something quite old. This was from a seminar at the Network of Aquaculture Centers in Asia, or NACA, in Bangkok in 1985. It's a training manual for the biology and culture of sea bass. This is not actually a document, it's a FAO website, and you can go in there and you can see the entire contents of the training class, scroll through the different pages and go through the table of contents, and you can learn quite a lot about how Asian sea bass are farmed in Southeast Asia. Here are some other papers that I can recommend. Aspects of the Biology and Culture of Lattice calcarifer by Mike Rimmer. He's an Australian. He's worked in Southeast Asia a long time. He was a consultant to NACA and he did a lot of training. So there's that. Then we have the Bioeconomics of Asian Sea Bass, Culture in Vietnam, and the Taxonomy, Identification, and Biology of Sea Bass. And the management of wild and cultured sea bass. This is from proceedings of a workshop held by the Australian government in 1986, published in 1987. And from Australia, the best source of information is actually this Barramundi Farming Handbook by Ship, uh, published in 2007. This one actually is um, sea bass culture in Thailand specifically. Once again, this is an FAO website. It's a training manual. It's available as an online document that you can read. And you see the information here. It was held in August 1988. And on the right-hand side, I've given you the chapters. And so it covers all kinds of things from larval rearing, hatcheries, hatchery design, broodstock, spawning, growing them in cages, growing them in ponds, managing the water quality, diseases, and so on. So this is also a bit old, but it's quite a useful website nevertheless. So there we are, part one of Asian sea bass. So please subscribe to my YouTube channel. Find me, subscribe in X. Find me in LinkedIn. And then like, comment, turn on notifications so that you can be updated when I release new material. And I'm particularly hoping that if you are a student or at a learning institution, that you will share this with your colleagues, your peers, share it with other students so that this information can be more widely distributed, because I'm really, really interested in having this information go out to new students of aquaculture, especially those who are interested in warm water species. So that's the end of this video, and I look forward to recording part two for you very shortly. So thank you very much.